Hi guys, this is Kumar. In this podcast, I'm going to talk about the structure of DNA. DNA, which is deoxyribonucleic acid. DNA and RNA are polynucleotide. Poly means many. That means both DNA and RNA are made up of many smaller molecules called as nucleotide. In case of DNA, it's made up of two strands of polynucleotide. So you can see here, this is one strand and this is the second strand. But in case of RNA, it's only one strand of polynucleotide which are attached to each other. Talking about the structure of DNA, each nucleotides are arranged into a ladder-like structure called as double helix. So double helix, they are kind of twisted ladder in which there are millions of tiny subunits of nucleotides attached to each other. Now let's look into more detail for DNA structure. Each nucleotide which makes DNA as a polynucleotide consists of three parts. 1. Phosphate group, 2. Pentose sugar, 3. Nitrogenous base. So the structure of nucleotide looks like this. On the top is the phosphate, and you are aware of phosphate from ATP. So you know in ATP, phosphate is the major part which releases energy after the breaking of the bond between the phosphate, and they release energy. So phosphate is attached to the pentose sugar, and this pentose sugar is attached to the nitrogenous base. So talking about nitrogenous base, we'll see there are 4 different types of nitrogenous base in the cases of nucleotides. So phosphate sugar and sugar are kind of a backbone of the DNA molecule where the base is from the rug. So if you look like this, these are kind of a backbone which are phosphate and sugar, and the middle part is the bases. So if you divide this into 2, you can say this is strand 1 and this is strand 2, which are kind of joined with each other in the middle to form a ladder with the rugs, that is the bases. So it's a kind of rungs which are running in between the backbone of these 2 strands. Talking about the nitrogenous bases, there are 4 types of nitrogenous bases which are part of the nucleotide. And the first one is adenine, which is represented by letter A, cytosine, which is represented by letter C, thymine, which is T, and guanine, which is G. So there are 4 different types of nucleotide. Guanine and adenine, they are called as purines, and cytosine and thymine, they are called pyrimidine. But there is a rule that each base will only bond with one another in a specific base. So adenine goes with thymine and cytosine forms with guanine. So they form a base pair, that is adenine is always going to pair with thymine and cytosine is always going to pair with guanine when they are making the structure of DNA. Let's look at this. So again, just repeating, this is phosphate, this is pentose sugar, and these are the nitrogenous bases. So adenine always base pair with thymine, cytosine is going to pair with guanine. But if you're looking at the strand, this is strand 1, this is strand 2. Now, there is position of carbon on these pentose sugar which are called as prime. So talking about this, when we start with the carbon, position of carbon on this sugar, this is 1 prime, 2 prime, 3 prime, 4 prime, and this is 5 prime. So the strands run opposite to each other. So if we are talking about the strand on your left, the 5 prime is up, which is attached with a phosphate. And when you're talking about the opposite strand, the 3 prime is up, and it goes from 3 prime to 5 prime, and this goes from 5 prime to 3 prime. So the 5 prime is always has the phosphate attached to that. So the second strand is opposite to that. So if the left hand strand is from 5 prime to 3 prime, then the opposite strand is going from 3 prime to 5 prime. So these prime are the positions of the carbon on the pentose sugar. So it's we start counting the first prime from the nitrogenous base. It goes to the second prime, then third prime, fourth prime, and the fifth prime is always attached to the phosphate. Now, because of this complementary bases pairing, the order of the bases in one strand determines the order of base in the other strand. So what does that mean? Let's have a look at this. So if it is the first strand and it has adenine attached to that, so in the complementary strand, the other base is going to be thymine, cytosine is going to be with guanine. If there is a thymine here in this strand, it's going to be adenine, and cytosine goes with guanine, adenine goes with thymine, and guanine goes with cytosine, and thymine goes with adenine. So in the other complementary strand, the bases are like that. Now they get attached, and that's how it makes the DNA. So DNA structure, to crack the genetic code found in DNA, we need to look at the sequence of the bases. So if we are talking about the sequence of the bases, going back to these are the sequence of the bases, like how it goes in DNA. And the bases are arranged in the triplets called as codons. So 3 bases, they make 1 codon. So this is 1 codon, this is another one. So this is how it goes for the genetic code. So summing the whole podcast into one slide, let's look into the cell. Inside the cell, the nucleus, there are present the chromosome. So chromosome are the structure which are inherited from your parents, and it makes you who you are, and are a set of instruction given by your parents. So chromosomes are present in the nucleus of the cell, and humans have 23 pairs of chromosomes. So these chromosomes are kind of condensed and are long strands of DNA. So if you uncoil it, it will form DNA. And this stretch of, when you stretch this, you remember on these DNA are the nitrogenous bases, which are having the codes for proteins. So each 3 nitrogenous bases, they make 1 codon. So one stretch of DNA, it makes genes. And these codes determine the shape and size of proteins, and the proteins make cells, cells make tissues, and tissues make your body, like your eyes and the skin. So genes determine what you are. So in this, we have learned about the structure of DNA and how it works for coding for proteins.